Okay, first I want to talk about X-inactivation in females, and then we'll talk about linked genes. So the thing we know about most chromosomes in humans, well, let's just use humans, is that you have two copies of each of them. You have two copies of chromosome number 1, two copies of number 2, two copies of number 3, all the autosomes. And that's why in each of the chromosomes, the homologs, each of them has a copy of the gene. That's why the genotypes we use are things like big A, little a, and little a, little a. It's indicating you have two copies of the A gene. So that way you get two copies of A gene expressed, which means you get a certain amount of protein being made from gene A. Let's say it encodes for the program that makes protein A. So you have, 2 times protein A, and that's normal. Males and females are the same. The thing that's different between male and female mammals, including humans, is that the female has 2 X's and the male only has 1. So this is different. Now the female would get 2 times let's say the H gene is on the female's chromosome. She'd get 2 times the H gene product, whereas the male would only get 1, because she has 2 copies of the gene versus the male's 1 copy. So what ends up happening is that we do X-inactivation, is what happens in female development. And so what this is going to do is to create what we call a bar body, which just basically means one of the female X chromosomes curls up really, really tightly and is not read anymore. None of the genes on it are transcribed, which means that they're not read to make protein. So what ends up happening is this doesn't happen right at birth. So all eggs start out, and the eggs in a female's body right now, they all have both their X chromosomes open. So they're both just like all the other chromosomes would be, not curled up. And the egg gets fertilized. Well, this would be a fertilized egg. So the egg would have 1 chromosome opened up. Then it would have gotten fertilized. The male would have brought this chromosome in. And as the female zygote is developing, it starts dividing. And then eventually, at one point in division, some of the cells inactivate 1 chromosome. Here they've inactivated the one with the orange. And some cells inactivate the other chromosome. Here's the one with the black. It's random which X's get inactivated in which cells. But every cell that's a descendant from this early developmental cell will end up only expressing the genes on the open chromosome. So this inactive one stays inactive. Even through every division, it still keeps on curling up after it divides. Any descendants from this cell are only going to express the genes on this chromosome, the orange gene. This, with the black gene, is going to stay curled up even through all the cell divisions. So that's how you get these tortoise shell cats as well as calico cats. They have other patterning on top of them, but the basic patchy color is due to this X-inactivation. So there's alleles for orange and alleles for black fur. And you can see that the female has, in this case, she's a heterozygote. But she inactivates, sometimes she inactivates the orange, so she'll get black patches, and sometimes she inactivates the black, so she'll get orange patches. Now I say she because these are 2 X's, right? So this is a female. This is the thing some people are familiar with, that you can only have female tortoiseshell cats, same with female calicos, and this is why. The way you get that pattern, which is why we call them tortoiseshell or calico, is by inactivating different X chromosomes during development. A male doesn't inactivate X chromosomes because he only has 1, so he expresses everything on it. So there could be either males are orange. If they get the X little b allele, they're orange. If they get the X big B allele, they're black. If a female gets heterozygous, X little b, X big B, she's tortoiseshell. So work that out for yourself in the book. So I would write down, the 2 types of genotypes males can have: X big B Y, X little b Y, and then figure out what those look like. Then there should be 3 genotypes for females: homozygous dominant, heterozygous, and homozygous recessive, all on the X chromosomes, and figure out what those look like. And then you can see why you could only get heterozygous females, which means you can only get tortoiseshell females. So of course, is unless something goes wrong in division and you get a Klinefelter cat, which has 2 X's and a Y. So then it'll make male body parts because of the Y, but it'll have 2 X's, so it can inactivate. Rare case though. Most tortoiseshells and calico cats are female. And let me show you this just real quick. I don't know if this records. Let's see. Sound for this part. Well, I know it'll do it here. Okay.
And these cells start off.
Sorry.
And these cells start off in a female with 2 X chromosomes. The cell divides, and the embryo is getting slightly bigger, and still each cell has both X chromosomes active. But in early embryogenesis, each cell will inactivate one of its X's, and one cell will remain with the paternal X as active, while the other one, the maternal one. And now this process will happen at random, and you'll have almost half of the cells with the maternal X active, half with the paternal X. The embryo will continue to grow, cells divide, all the descendants of that cell, all the descendants will keep the same X active. And you've seen this in calico cats, right? The coat color gene is on the X chromosome, and that's precisely why these cats look like this, a mosaic appearance, because of the process of X chromosome inactivation.
So there you go. It's a nice little animation for you to see that. Okay, so that's X-inactivation. Then I want to talk about X-linked, or not X, linked genes. So this is different than what we've been talking about. We were talking about X-linked alleles, so genes on the X chromosome. Now we're just talking about what we call linked genes, which means genes on the same chromosome that tend to be inherited together. So basically this means they're really near each other, and so they travel together. I'm not going to go through this entire example, it's in the book, but the basic idea here is that you had a parent that was completely wild type, the fly, so that means it has red eyes and a gray body. And you have another parent that's completely mutant. Oh, excuse me, it's not eyes. It has a gray body and normal wings. In this fly, this is completely mutant, it has a black body and these little stubby wings. We call them vestigial wings. You cross them, and then you know you get a heterozygote for both of the genes. You recross it you do, with your recessive, because you know that's not going to cover anything up. And if you do our normal Punnett square, we're expecting to get about a quarter of each of these types, right? Sometimes our gray body and our regular wings will be together. Sometimes our black body and our vestigial wings. Sometimes our gray body and our vestigial wings. And sometimes our black body and our regular wings. So we expect them to mix up. But what happened is that he actually saw different ratios. So he saw that there were about 900, 1,000 of these gray normal winged and the black vestigial winged. And then he had very few, comparatively, about 200, of the gray vestigial and black normal. So what you're seeing here is you're seeing that the parent, the two that the parent looks like, get the most offspring. Whereas the ones where you're mixing up two of the parent's phenotypes, right? The gray comes from this female, the vestigial would come from the male, and then the opposite here. When you're mixing them up, you don't get very many of those. So this means something else is going on, to where these are not randomly getting put into eggs and sperm. It means you don't make very many eggs and sperm that look like this, the combos. Okay? So here's what he figured out is happening. So the female, we know she's heterozygote for both of the genes to start with. And turns out he found out that they're on the same chromosome, and you can see why this is. So if he has the two, we call them wild type, the two normal versions of the genes together, and the two mutant versions, that's these guys. So the two normal ones are going to travel together, right? And the two mutant ones from the female travel together. And since the male has all mutant, which is recessive genotype, he doesn't cover up anything. So we're really just looking at what does the female give. So this female, in this case, she gives both wild types. So that's the gray body and the straight normal wings. And then on this chromosome, she gives her mutant version. So her black body and her vestigial wing chromosome. So you can see here they stick together. So then that's when you get these different ratios, where you get mostly parental phenotypes. So your offspring mostly look like the parents. They don't get mixed up. And that's what he sees here. We call, so you did notice though, that it's not 100% not mixing, right? So even though they're on the same chromosome, and they should move around together, sometimes they don't. Sometimes they still get mixed. And so how that happens, we can think of the different parts of meiosis we know of. That's when we're making these eggs and sperm and mixing all the genes around, right? We're trying to create genetic variation. One of the very first step we do that in is when we do crossing over in prophase 1. So I will show you here what I'm talking about. So here I have two chromosomes. One was from this going to be the mom. So the mom's dad gave her all big letters, big A, big B, and big D. And then her mother had given her all little letters, little a, little b, little d. So this is the mom. And the mom is going to go and she's going to make some eggs. So the first thing that happens is that the chromosomes get replicated, right? Exactly the same genes in exactly the same spots because their sister chromatids are identical. You notice that these two chromosomes are homologs, right? Same genes in the same spots. They could have the same version. These have all different versions. So if we looked at mom's genotype here, she would be big A, big B, big D, little d. So you remember here what we usually do in prophase 1 is we do crossing over, right? So here's some crossing over. Now what you'll notice is that because our A's and B's are close together, most of the time they're not going to get pulled apart when they cross over. And by that I mean we're not going to cross over in between them. You see, for me to cross over in between them, I have to aim very, very specifically right between the two. And that doesn't happen that frequently. Most of the time the crossing over is going to be going on somewhere where there's lots of room. Here, 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 something like that. So because they're so close together, they're really hard to separate. You'd have to aim. Of course, it's all random. Well, not completely random, but the probability of crossing over between two closely spaced genes is low. On the other hand, you see here that we have our D gene, it's far away. And so if we can imagine what's going to happen here, we're going to have our ends change, right? So we're going to exchange, we're going to exchange our ends. So in the end, we'll get a blue version on this side, right? Something like that, you can tell what I mean. And then we'll get the same thing, but it'll be the red version when we switch. Now the thing is, we're not just switching parts, we switch the genes on them too, right? So this would be now on this chromosome, and that would be a red color. But we switched the genes as well, we didn't show that. So now we took the big D, let's see if I can move it, oops. Do it this way. we took the big D and we switched it, right? Okay, with the little d. Okay, right, which makes sense because our little d was on the red and now it's going to be on the blue. So you can see when this separates, this whole arm's going to go together. So now the big A, the big B, and this little d are together. The little a, the little b, and this big D are together. So this crossing over mixes it up. And because the A and the B are far away from the D, this happens more frequently. You see there's a lot of different space that they could cross over in order to mix up those genes. So the further away on a chromosome the genes are, the more what we call recombination there is. So the frequency of recombination, so we call that the recombination frequency, recombination frequency is very low between A and B because they're close together. The recombination frequency is higher between B and D, and it would even be higher between A and D because they're even further apart. So the further apart they are, the more they recombine. And then we have the scenario where we actually have chromosomes or genes on separate chromosomes, right? This is mostly what we were talking about before, where it's completely unlinked. So when it's completely unlinked, we know that we can line up in two ways, right? We can line up like this with our blues on the left and our reds on the right. So this would mean that the big H would segregate to the left side, along with these big letters here. The little h would segregate to the right side and have these little letters. So then we'd get a cell over here. The egg would be little a, little b, little d, little h. And this egg would be big A, big B, big D, big H. But we know from independent assortment that there's no good reason that it has to line up like that. It could line up the opposite way. And now you can see our big letters segregate with the little h, and our little letters segregate with the big H. There's no reason that can't happen. So it's a 50-50 chance. So in metaphase 1, how they line up, how they independently assort, is 50-50. So if you see a 50-50 recombination frequency, they're probably on separate chromosomes, or they're really far apart on the same chromosome, and basically half the time cross over, half the time they don't. So those are not linked. A 50-50, 50% recombination means they're not linked, because this is what's accounting for it, how you line up. If you see very small, like this would be like a 2% recombination frequency, they're really, really close. Between A and D would be more like a, 35% recombination frequency. And then maybe between B and D would be more like a 28% frequency. So you can see how that works. So that's important because that's why certain traits travel together, right? Some traits travel together because they're actually encoded by the same gene. So that's one option. The other option is that they're linked. So here we can see that there are certain genes linked in these P's in this case, that we get mostly parental, but we also get some recombinant. Actually, in the P's, they were 50% each, right? Which we knew because when we it's 50% each, so that means these are unlinked. Now, if there were more of these parental and less of the recombinant, then we know that they were linked genes. So that's what this is showing here, basically. Sometimes you get recombination, sometimes you don't, and so you get all these different combinations. And then this ability to calculate recombination frequency is important. So it basically makes logical sense. If you want to know how frequently something recombines, you want to figure out what percentage of your offspring are recombinants. So you're going to take the numbers of everybody that's mixed up, all the mixed phenotypes that are different from the parents, that are a combination of two. You take those and you add them together, and you put it over the total number of offspring. So it's strictly the percentage of recombinants. That's why we multiply by 100. So the percentage of the offspring that have been mixed up from the parents, that is called your recombination frequency. So, like I was saying, if they're very far apart, here's like a 17, they're going to be much longer. Recombination frequency is bigger than if they're closer together, like a 9. These are also, you won't have to order any kind of genes, but I'll just show you that, like the recombination frequency between short aristae, these are the little antennae things, and the long, so this is the two versions, these are the two alleles, long versus short. And way far away is the brown eyes versus red eyes allele, or gene, the two alleles of that gene. 104.5, so they're really far away distance. So these would have a very high recombination frequency. So frequently you'd get the aristae that are short mixed with red eyes and the brown eyes mixed with long aristae. On the other hand, gray or black body travels with cinnabar, these kind of orange, really orange eyes, really travel together most of the time. And gray bodies tend to travel with the red eyes because these guys are close together. So they don't have crossing over happening between them very frequently. Okay? And then the last kind of concept I want to just really touch on here, we talked about this in class already. It's called nondisjunction. I just want you not to forget about that. And this is a prettier picture than I could draw, but here's the idea, is that you get chromosomes accidentally segregating improperly in meiosis 1. You could have it in meiosis 2, so either one. In meiosis 1, that means you end up with 3 copies, or 3 chromosomes here and only 1 chromosome there. And then in the end, your eggs, or your sperm, gametes, these have 1 extra, right? So it now has 2 of the big allele, or the 2 of the big chromosome and 1 little. And sometimes they're missing. This one's missing the big chromosome completely. So these are all problematic from this nondisjunction in meiosis 1. In meiosis 2, we don't even expect this to happen, but if it does, it's very rare. So only one of our 2 cells is going to have it happen. So this cell goes on and it's normal, where it has the normal N haploid number of chromosomes, 1 big and 1 little. We have nondisjunction on the second round of the anaphase 2, and now we have 2 copies of the big and 1 little, and 0 bigs and 1 little here. So we're missing 1, and we have an additional 1. And these are the kinds of things that lead to what we call aneuploidy. So when you have 3 copies of a chromosome, so that would be if you got an egg or sperm like this, typically the egg, where it has +1, because let's say this is chromosome 21. So now mom is donating 2 copies of 21. Dad comes in and donates 1 more copy, like he usually does, and you have 3 copies of 21. So you can look at that here on this karyotype. There are also some karyotypes online, a PowerPoint slideshow you should check out so you can recognize different karyotypes. You'll also need that for your lab practical. So here's the Down syndrome, or trisomy 21. Tri meaning 3 21s. Frequency increases with mother's age, past 40 particularly, exponentially increases even within the 10-year span from 40 to 50. So that's why they encourage women to have children before that age, because the probability of these nondisjunction events happening is very high. Because if you recall, females, even though they have all their eggs made when they're fetus, they don't actually finish meiosis until they're released and actually fertilized. So you still can make mistakes. Also, if you were to get 2 X's in one of the cells, or an X and a Y in one of the sperm. So by accident, the egg gets 2 X's, or the sperm gets an X and a Y. Then you get Klinefelter syndrome. If you choose one of these eggs that's missing a chromosome, so now there's no big chromosome. If it's the X chromosome, you'll only end up with one, right? So dad gave you none, and mom gave you one. So this is called monosomy X. That's Turner syndrome. You'll notice there's not very many of these. Most are non-viable, which means the fetus or the embryo, depending on the stage, usually embryonic stages, they die very early because they can't, there's so much problems with having extra or missing chromosomes that the organism can't actually develop. I have to mention this for evolution. So polyploidy is where you copy the entire set of genome, the entire genome. So instead of just having one extra, chromosome 21, you have one extra of everything. So if you were to look at a karyotype, there would be 3 of every type of chromosome. 3 1s, 3 2s. That's triploid. Tetraploid would be when you have 4 copies of every chromosome type. So you have 4 number 1s, 4 number 2s, 4 number 3s. This is more common in plants. And this kind of duplication of a whole set of chromosomes can lead to evolution because you have another set to kind of play around with without making the organism die. It's kind of how I think of it. So if mutations occur, sometimes if they occur in an organism, then, and it's, there's a non-functional gene, that organism doesn't survive. But now if you have 4 copies of that gene, and you have a mutation in one of them, you might be okay. And that mutation could, in a different environment or in combination with other mutations, could become advantageous. And so then you go down this little path where you have extra copies of genes and eventually could, become a new species or at least evolve to adapt to the environment. So the polyploidy is particularly important in plants, and particularly important for having excess genetic material in order to accumulate mutations, which can be a good thing, to allow you to evolve. And that is one of the few mammals that I know of. I don't actually know what it is, but it's a little mammal that's a tetraploid mammal, has 4 copies of every chromosome. These are very self-explanatory. Look at these pictures in your book and just understand what they would be. These are chunks of chromosomes. So when chunks of chromosomes are lost or flipped or repeated, these are the different kinds of gross chromosomal abnormalities that can occur. Okay, so just be familiar with these deletion, duplication, inversion, and reciprocal translocation. Kind of like crossing over, but not, because you exchange different chunks of chromosomes, different genes. That's not good. Okay. Don't worry about imprinting. Don't worry about that for this exam. All right. So that's basically what I wanted to tell you about linked genes and a couple other little things. I may post another video just about genetic testing. It'll be very short, much shorter than this one. Okay. So I hope that was helpful. Let me know if you have any questions.